All right, genetic mutations. I wonder how many videos I've started by saying, all right. If you're watching them all, probably every one, eh? Because, I don't know, how else do you introduce this? How do you start? It's weird. Good morning, everyone. Doesn't make sense. So here we go. We're going to just basically look at what a mutation is. And essentially, a mutation is a change in the nitrogenous base sequence on the DNA molecule. Because the sequences of bases are the codes that spell out how to make proteins, then if you change the order or the sequence of those bases, if you have a C where a G should be, or if one gets deleted, or if something gets added by mistake, that means that the words change. And it could mean something small, but it could also mean something big, depending on what the change does and how significant it is, and whether or not it changes something that's essential or something that's just kind of, mildly handy to have around, right? All the proteins and things that we do. So you have to remember that the DNA molecule, like any molecule, it isn't a solid structure. Remember that at the base, everything is made of atoms that are sort of bound together by this desire to keep their electrons in a certain way. And so what that means is that everything is sort of held by these bonds, some of which are strong and some are not so strong, and they can easily be bumped and broken and shifted by the interactions of other molecules. So a molecule, or I guess we could say a molecule, or I don't know what a word would be, a phenomenon. That's not a very good word. I'm trying to think of another word because there are things like ultraviolet light and things that can do this. So a molecule, or all right, phenomenon. That's all I can think of. That can change the base sequence. Is called a mutagen. Mutagen. So a mutagen, it causes. To generate means to make or to start or to cause. A mutagen generates a mutation or causes a mutation. And you may have heard of the word carcinogen, and that's the same idea. That's a specific mutation that actually results in a cell becoming cancerous. But there are other mutations that can do other things. So a carcinogen is a very specific kind of mutagen. Right? We'll talk about how cancers work later. Okay, so we know what a mutation and a mutagen is. Now all we have to do is sort of look at some of the things that can happen and some of the ways that these mutations can take place. Okay, so we're just going to make a list of all the possible things that can happen. First, we're going to talk about, so let's put this down as a chart or something, types of mutations. Then you can have a handy reference. And I'll bring this up so we can see it. Are you okay with that? In your textbook, this can be found on page 340 and 341. And there's actually a nice handy chart there, what we use later. Okay, so there's what's called a point mutation. And this is a change in one nucleotide. So one spot on the DNA. Remember that a nucleotide is basically the nitrogenous base, the ribose sugar, and the phosphate group. So those things get sort of knocked out of place. And if we were, imagining this, our good DNA would have perhaps C, well, it looks like I made it a G, GCCTA. And a point mutation would show up GCGTA. And so we take our black here and we circle a single spot where a base has been changed. So that's called a point mutation when that happens. Okay. This is also sometimes referred to as a substitution. So basically what we're doing is we're going to see all these words that are sort of related to each other. Now, when I say a change in the nucleotide, this particular change I've drawn is what we would call a substitution, where one base is changed to another. Substitution. But it could be that it's not a substitution. It could just be a rendering of unreadability. So, for instance, it could just be this. It could be, the GCCTA, and on the new gene, a molecule has interfered with this, so now the C that was normally there has been sort of interacted with, it's been changed, some of the electrons have been sort of taken away from it, right? So maybe that C is no longer readable. Maybe when the RNA polymerase is trying to scan this gene to create the messenger RNA strand, it can't read that for some reason. So a point mutation just means at a single nucleotide spot, but it could be a substitution. It could be what we call just sort of an unreadable thing. It could be what we call an insertion. This can happen at one spot. An insertion would be when you add a base in. So an insertion would be GCCTA as the normal DNA strand, but somehow we get GCCTTA, and we can see here that we have an extra T that's been inserted in. Right there. That's the insertion. So now, of course, that screws up the codons. Right? If you were reading these codons, in fact, if I add one more letter here, let's add one more G on the end in both colors. You can see the codons originally. Here's the first codon, here's the second codon. Well, now when you read this, you have a codon here, which looks the same, but the second codon is now read as TTA instead of TAG, which would code for an entirely different amino acid, or perhaps nothing at all. And that would create a problem. So what that actually does is it shifts the frame of reading the codons, right? Insertions. You could have a deletion, right? A deletion. So we could have G, C, C, T, A, G. We'll use a 2-codon sequence from now on just to show it how it works. Well, you get G, C, PAG, and again, you can see that in this particular spot between there's a C missing here. It's been dislodged. That's called a deletion. Are we going too fast? Okay, so there was the deletion. You're going to have something called an inversion. So let's talk about an inversion. Go. An inversion. I have to draw the double strand to show you this one. So we'll have a strand here, and we'll have our, on the bottom, we'll put our GCCTAG again that we've been sort of using as our standard. Then what will happen on top, we can predict what will be CGGATC, right? That's the complementary strand of the DNA. And in the inversion, what happens is there's a flip. So in the new DNA, we would get G, C, C, and then the A that was on top would flip down. And the T that was on the bottom would flip up. So these two things, because they're sitting there together, A, T would go up. T, T, C. So I'll circle that spot so we can see it. The T and the A sequence have been flipped upside down. Now that makes an important change because only one side of this DNA is going to be read by the RNA polymerase to make the messenger RNA. So if it was reading the original bottom strand, the RNA strand that corresponds to T would be A, and it would build an A in that spot on the messenger RNA, right? But now when it reads this one, it's going to put a U in that spot, which changes the code automatically, and now you have a different situation. You learned this already? Did you learn this last year? Okay. Okay, that's good. Now we can have mutations, sort of the effects of mutations. So those are sort of the different kinds of point mutations that you can have, right? Point mutations. But there's also sort of broader scale mutations. So I guess what we would say is the effect of the mutations. Let's call it effects of mutations. Now, what you have to remember is that not all of the bases on the DNA are actually part of the genetic sequence that's coded for proteins. You remember the introns. They're the extra little bases that aren't really important for anything in terms of the expression of the gene. And so if these mutations happen at spots that are not critical, if they happen to the introns, then nothing really matters. It's only when they happen to the exons, the points that are being, the points of the DNA that are actually being copied for the messenger RNA to make a protein. So the first sort of effect is what we call a missense mutation. Let me go back to my missense mutation. And this means that the mutation results in a different amino acid. Being placed in the polypeptide chain. Okay? So when we're talking here, it could be any one of those other mutations that happen. It could be an inversion, it could be a whatever. But when the result is that a new amino acid is coded for because of that point mutation, we call it a missense. So let's actually do a diagram of what that might look like. All right, so we're going to look at our normal DNA. And your textbook has some pictures like this too. I'm just going to draw sort of smaller versions of it. So if we take a piece of normal DNA, and I choose 2 codons here. So let's say we go ACC, GGT, actually I'll do 3, and TCA. Let's say that's the strand. Then the complementary strand would be TGG, CCA, and then AGT. So there's our normal piece of DNA. Now, for all of these, we're going to use the same normal piece. So you can keep coming back to this to see what the normal one would have looked like. Now, we can actually look up what's being coded for here. What we'll say at the bottom strand is the one that's being coded. It's the 3 to 5 direction, let's say. So those 3 amino acids, if we go back to the table, we can find, let me just go back there and get the amino acid list, and we can see what those amino acids are supposed to produce. Right? Where did it go? Now I can't find the the page. Here we go. So it looks like TGG on this bottom strand would form, oh, we have to remember that the codes are in RNA language. So the RNA would be UGG, right? Because it has a U instead of a T. So the RNA, well, actually, no. The RNA would code off of that, and it would be UCC, wouldn't it? No, it would be ACC if the RNA coded off of that. It has to build complementary. So if we look at that as ACC, that would be a threonine, I think. So this first amino acid is threonine. I'll just put TR like that, threonine. And then the next one should be glycine. Glycine. And then the next one should be serine. Right? So if you're reading a DNA piece, you have to imagine what RNA would code off the bottom, ACC. Look up ACC, and you'll find in the chart that ACC is threonine. CCA on the bottom would code for GGU on an RNA. And if you look up GGU, you'll see that GGU is, let me see if I can find it, glycine. So that's how you do the coding, right? That's how you read the code. You gotta make sure you know if you're reading DNA or RNA, because the codons are always in the RNA language, right? Okay, so let's draw beside it a mutation then. So I've used red for, I'm going to change my colors. I'll use green then for the mutated version. Let's draw the mutated version here. So in the mutated version, we would have ACC, GGT, and then we're going to have a mutation here and put CCA. So that's a point mutation. Down below then, we would have we would have T, G, G, C, C, A, and then G, G, T. So our third codon has been changed. I'll circle the change so we can focus on it. And it's been changed right here. This section has been changed. So then what happens is if you read the code, right, you'll get the threonine still coded for, the glycine is still coded for, but now instead of an RNA reading UCA, it's going to read CCA, isn't it? And if you look up CCA, let's go to that one, that's proline. So what we've done is we've turned that amino acid serine into a proline. We've changed the amino acid that's being made, and that's going to make an entirely different polypeptide chain, which will take on an entirely different secondary and tertiary and quaternary structure, because proline will bend and twist the molecule differently than serine did, and the shape of that molecule will now make it either dysfunctional, or it'll perform a completely different function altogether. So there you go. A missense is when you get a new amino acid in the chain. Okay? Fair enough? Pause? Okay, let's carry on. I'll draw a line here. Go back to black. All right. The next one we'll do is what's called a nonsense mutation. In missense, you can understand it. You're misreading because you're reading a different word. Okay? Nonsense means it just gets screwed up. So a nonsense mutation occurs when a point mutation causes something else to happen. It causes a premature stop code. Remember that some of the amino acid code words are not for amino acids. Some of the codons are for stop and start. So if you get a normal one that was supposed to be an amino acid, but it's changed into a stop, well, that's going to stop very prematurely, right? So if we do our little thing again, I'm just going to go down here a bit so I can see my normal DNA, right? So we'll draw our normal DNA piece again. I'm going to point mutation. Oh, causes. Sorry, I forgot to write the word causes in there. Causes. Insert causes, a premature stop codon. All right. So ACC, we can go faster now because we've got it sort of figured out. GGT, TCA, normal DNA, TGGCCA, AGT, which again codes for threonine. Not sure. I'm looking up here on the other one. I think my brain was already thinking of glycine, and I put a Y there. Threonine has got an E. It doesn't have a Y. So threonine, let's just fix that for good looks. Glycine and serine. But let's draw in green our mutation. So for the nonsense, here's what we're going to want. We're going to want sorry? Serine. Yeah, you can look those up in the chart if you're not sure. Okay. Now, in the textbook, of course, they decided to move this to another spot. So I actually have to add another sequence because the TCA won't do it. Stupid book. Well, that's a good question. So if you're trying to figure out where the codons come from, you have to remember that the RNA polymerase rides in which direction? 3 to 5. It builds 5 to 3, the RNA, but it rides. So if we would have to specify on here, but this is the 3' end of the bottom strand, and this is the 5'. So we're just assuming that we're always using the bottom strand in these examples. So the top strand isn't what the codons of messenger RNA would form from, okay? So what I have to do is I have to add another codon, because this one has to be specific, because we have to make it spell stop. Okay, so I'm going to add one more codon after the serine. After the TCA, I'm going to add TGA. So on the bottom, we would have ACT. That's the normal DNA, I think. Oh, that's the mutation. Sorry, TCA. I got to line this up. Not TGA, it'll be TGC in the normal DNA, right? It'll be the, sorry about that. There's two different pictures and I'm confusing them. So let's go TGC right here, and ACG, that's the normal. And so normally that would be cysteine. So the normal thing here would be cysteine. That would be made there. But cysteine can easily be turned into a stop codon by a single mutation. So let me just use a shorter version, and I'll start at the serine. I'm going to start it right here rather than draw all that again. TCA, and this is where we get TG, but this time it'll be mutated to TGA. And then AGT, ACT, right? So there's the mutation. This last little code here has been changed. And then ACT on the bottom, if you think about the RNA version of that, it would be building a UGA. And if you go back, you'll find that UGA is a stop code. So UGA actually spells stop. So now what we have is we have the serine from before, but this is spelling stop. Now this is a serious mutation because there could be a great deal of the gene behind here still waiting to be expressed. In this case, this nonsense mutation means that the entire polypeptide chain would be truncated at that spot. And that would mean the protein is nowhere near what it's supposed to be. So this is far more serious. It could basically mean the protein is completely dysfunctional, right? It's like a third or a quarter or a half of what it should be. So this is the nonsense mutation, and it results in a stop codon instead of a change in the sequence of amino acids. All right, so our next is called the silent mutation. And this is what happens when a part of the DNA code that is not coding for a particular protein is actually changed in some way. So it's still a mutation technically, but it has no real overall effect because it's a piece of the DNA that isn't coded. Okay? Or, well, let's write that down. It's either a change in the portion of DNA that is not part of the code, of the enzyme code. So you'll remember the introns and things. Or remember that there's redundancy built into the system. Or the mutation produces a new code for the same amino acid. Because remember, some of the amino acids have more than one code, right? So we can do a picture of that one just to show you what it would look like. I'll just go up a bit here and bring it back down for you. Let me see if I can find an example of that. Or, actually, what it should be is the comma should be after the or, right? Grammatically speaking. Or the mutation produces a new code for the same acid. Got it? Okay, so if we do this one, I'll try to get this from if we have, let's look at the code ACC, or the DNA that goes ACC. And I'm just going to do the next one over, which would be GGT. There was our original DNA, and that would mean we'd have TGG here, and we would have CCA on the bottom. So let's change one of those. Let's change the the GGT. Let's change it to ACC, and then let's say that the GGT gets changed to GGC. That would make this TGG and CCG on the bottom. All right. So here is where we've made the change on the original DNA. And if you recall, on the original DNA, the ACC part was the threonine code, and then the glycine code. So over here, what do we get? Well, over here, if we read the codes, we've got the threonine. The new CCG code would produce a messenger RNA that would be GGA. The original one was GGT. But I think, if we go back to the chart, we'll find that GGA is also a code for glycine. I'm pretty sure that it is. So this CCG section is now producing also a glycine again. So even though there was a mutation, nothing has changed in the amino acid sequence because there is more than one code for some of the amino acids. right? Makes sense. So there's two things that can happen for a silent. It could be part of the enzyme or part of the DNA that's not actually in the exon, or sorry, not the exon. Well, yeah, it's not in the exon or it's not in the operon of the gene. It's just that intron extra piece that gets cut out anyway by the splicing process, right? It gets cut out and taken away from the whole process anyway when we make the messenger RNA. Or it could be making the same amino acid. And we have one left to discuss, and this would be a frame shift mutation. This is also a serious thing. Frame shift mutation. What happens here is the change in the DNA completely changes the way we read the three-letter codes. So this usually happens with an addition or a deletion of a base. Because if you add one or take one out, all the three-letter codes get pushed over, and they're all read wrong. So the entire thing is all wrong because of the frame shift. So let's write here. Let me get a smaller. The insertion or deletion of a base or a nucleotide. We could use the word nucleotide, which we probably should, but I'm going to put that in brackets because the nucleotide is actually the whole piece gets pulled in or out. So the insertion or deletion of a base or nucleotide results in a shift that causes all the following codons to be misread. In other words, they're all going to spell different words now. And that means that they might spell stops, they might spell new amino acids, there could be a whole bunch of different effects. So if we just draw one more little picture, and really this one is an easy thing to do. Let's draw our original DNA here. And let's again start with our threonine. So we have the ACC on top. Then we have a G. GT was our original that we had. And then we have a CTC, I believe. Actually, I'm going to draw one more here just to show you. Oh, TCA. Sorry, you are right. Be careful to look at the right one. So G, G, T. Okay, TCA, TGC. Okay, you can see it, I can't. Good. So that was the original piece, right? So then we have the T, G, G, C, C, A, G, T, A, C, G on the bottom. Okay. Now our warped or mutated piece is going to look like this. There's going to be an extra letter in there. So in our mutated piece, we're going to have the ACC still there, but now we're going to insert an A. So G, A, C. Nope, GAG, sorry. So right at this spot, right here, we are inserting this A. And then the rest will follow, T, right? And then T, C, A, T, G, C. And on the bottom, we'll have T, G, G, C, T, C, A, G, T, A, C, G. Now the problem is with the insert of this A and T right here at this point, we now have different codons, right? Yeah, that doesn't matter. So here we still have, if we look at the way the codons are framed, here's the first codon, the second codon, the third codon, and the fourth codon. Up to here, everything's okay. So we'd still get our threonine, I believe that was. But now the next codon is read as GAG instead of GGT. And the next is this and this. So if we're looking at the bottom of those frames, the TGG still codes for threonine, but the new CTC on the bottom, right here, in this frame, this new frame, is going to code for, I think, glucine. Or glutamine, sorry, glutamine, not glucine, glutamine. And then looking at this one, it's a new AAG, and that's going to code for leucine. And then this one's going to code for methionine, I think. And so on. So everything's out of order because it was supposed to be threonine, right? And after threonine was supposed to be glycine, but it's no longer that, it's this glutamate one. And then you have AGT, that was supposed to be serine, I think. Now it's leucine, and it was supposed to be cysteine, I think, after that. And now it's methionine, and so on. So basically, they're all messed up. So this is a huge, huge problem. The biggest problems of these are the nonsense, which puts in a stop code. That's a big, big mutation. Or the excuse me, the frame shift. If that happens, that pretty much means whatever protein was being made is now gone. It's defunct. It's completely messed up. The other ones may or may not be as bad. Sometimes all it takes, though, is one change to make the whole thing inoperable or dysfunctional. But you can see how they're not that bad. Now, the other thing is, don't forget, your DNA does have the ability to repair itself. DNA polymerase II, right, is one of those repair enzymes that can scan along the DNA and find some of these mistakes and fix them. But it would be much harder to fix a frame shift than it would to fix any of the others. Even the stop codon could be fixed fairly easily. All you got to do is change that one base, right? Where was that? Right here. From stop to whatever it's supposed to cysteine and put it back. It's still a one base change. To do this, it's still a one base change, I guess, but you'd have to delete the AT and get that out of there. Okay, so that's basically all there is to understanding those. And all we want to do is be able to identify sort of what we mean by each one, right? And some of these effects that can be caused by it, the frame shift, the silent, and so on.